Hello, everyone. If you're new to our webinar series, welcome and thank you for joining us. Criterion Edge runs multiple informative webinars throughout the year. For today's webinar topic, we're talking about how to establish the competitive landscape within the scientific validity report. Our speaker today is Dr. Sarah Chavez. She is a principal medical writer with Criterion Edge. Sarah has a BS in biochemistry and a PhD in molecular and cellular biology with 25 years of laboratory bench experience and has taught at the university level for the past 15 years. She has extensive scientific writing experience in an academic environment as well as for several large textbook publishers. Sarah's strong background in basic research and her extensive experience with regulatory writing will allow her to assist companies with IVDR readiness as part of the Criterion Edge team. And I'll turn it over to you, Sarah, to talk about our presentation today.
Hey, thank you, Olivia, and thank you to everybody who's joining us today. During this session, we will be reviewing the scope and the purpose of the Scientific Validity Report, which I will refer to as the SVR. In particular, we're going to be discussing how the SVR helps you to establish that your IVD can be considered state-of-the-art. So our goal is that by the end of this presentation, you will all have a better understanding of why the SVR must include a clear discussion of the state-of-the-art, or the SOA, where to identify relevant performance objectives for your IVD, what data can and should be included in the competitive landscape, when to include detailed screening and appraisals of the articles that you've identified, and finally, how to present the data to determine the acceptability range. So as many of you are already familiar, SVR templates and any template will certainly vary, and it should be customized based on the unique needs of your IVD. However, IVDR is quite specific in defining what they consider to be scientific validity, and that definition is that scientific validity is the association of an analyte with a clinical condition or a physiological state. So I think it's really helpful to keep that definition in mind as you develop the outline for your SVR. There are some important references and feeder documents, such as your IFU, that can help you get started. But today, we're going to be discussing how the SVR relies heavily on defining the state of the art. So that's where having experience with MDR can be very helpful. While the SVR is a new requirement, the approach we're going to take to establish SOA in an IVDR world is essentially the same approach you would take if you were writing the SOA section for a medical device in a CER. The first thing to point out, if this is your first time planning an SOA section, is that you should expect to conduct multiple systematic literature reviews. This is not just a one and done quick PubMed search. It's a very detailed, organized, and a scrutinized process, so don't try to rush it. You will want to go into this process with a very clear plan and a solid understanding of what the notified body expects to see as part of your final report. The articles that you are going to retrieve in the first systematic literature review I've listed here, the SOA, will provide the information relevant to the background of the SOA section, which includes things like the medical condition or the patient population, as well as diagnostic alternatives and the risks and benefits of clinical testing. We discussed this first part of the SOA in a previous webinar, so I really encourage you to check that webinar out if you missed it. So today we're going to focus on the second systematic literature review I've listed here, which supports the competitive landscape. In MDR, this section is sometimes referred to as the maturity of the technology, because this is where you will want to go into detail about the IVDs that can be considered to be the most similar competitors for your IVD. This data will be appraised and extracted to present your competitive landscape. So you really need to have a solid sense of not only your competitors, but the measures by which you can compare the performance of these competitors. In our webinar today, we're going to walk through the SOA process from start to finish. So we'll try to show you how these two independent systematic literature reviews will ultimately help you tell the story of your IVD in the context of scientific validity. But before we get too far ahead of ourselves, let's take a step back and think about the purpose of the SVR again. First and foremost, do you understand why establishing scientific validity requires defining what we mean by state of the art? So according to MedDev Rev 4, the state of the art describes what is currently and generally considered standard of care or best practice for the medical condition or treatment for which the device is used. If we apply the same definition to IVDR, the SOA can be thought of as what is generally considered to be the standard of care for the diagnosis of a medical condition or a state. The state of the art does not necessarily imply that your IVD must be the most technologically advanced solution, but it should include a discussion of the medical condition, the alternative diagnostic tools, any relevant clinical guidelines or recommendations, as well as the risks and benefits associated with the use of this test to make a diagnosis. In addition, the SVR must also explain the association of the analyte and the medical condition to make sure that that connection is very clear. These are all critical components of the SOA section, and that information will be gathered in large part from articles that you've retrieved through your SOA search. You're also going to want to include a pretty detailed discussion of current recommendations and guidelines related to clinical diagnostic testing. So that will likely include articles that you've retrieved through your SOA search, but it could also require additional ad hoc searches to help you identify statements that have been published from experts or organizations. Now, this can be really straightforward if your analyte has a well-established association with a particular clinical condition, or if it has a really long publication history. But suppose your IVD is designed to detect a novel analyte, or if there's limited evidence regarding your analyte in published literature. In this case, you're still expected to present alternatives or discuss current clinical guidelines. But you'll need to carefully consider the strategy necessary to establish scientific validity. And that's why it's really critical that you begin your scoping process early in the preparations for your PER as a whole, because the limited data supporting scientific validity here in this report will significantly impact the quality of your PER as a whole. And finally, that's where we get to the competitive landscape. The competitors are different than alternatives, so keep that in mind. This is the section where you're going to be establishing safety and performance measures, as well as your acceptance criteria. And you're going to use data that's been extracted from articles retrieved through your competitor systematic literature review. The goal is to compare your IVD to the competitor IVDs to show that your IVD performs as intended and meets these established acceptance criteria. You'll also be expected to document each of these searches, particularly that competitor search, in detail and present your results clearly. The process of building out this SVR template relies heavily on the results of your systematic literature reviews. But don't forget that this is a process, and the entire process must be objective, reproducible, and well documented. The notified body expects to see that all relevant articles that you've identified in your search were screened and appraised in a very transparent, clearly defined manner. There are the two different SLRs here, which we will discuss in additional detail later in the presentation. The information that's contained within the SOA is critical not only to the SVR, but also will directly impact your CPR and your APR. So this is really where I like to point out that the SOA is that connection. It's helping to pull these reports together to truly tell the full story of your IVD, because we know that the SOA has to provide the background and the context for that clinical connection. That part's pretty clear. But we're also trying to set the standards by which we can measure our IVD relative to the competitive landscape. So now what? Now we need to know what are our clear safety and performance objectives. But how do you make sure that you've actually selected the best ones for your particular IVD? Based on our experience with CERs, a critical first step is to fully identify your intended clinical benefits. These are not just commonly reported endpoints, and in our experience, that seems to be one of the major points of confusion. Clinical benefits can be and should be informed by guidelines and recommendations and medical standards, but they also must be meaningful within your own quality management systems. While direct clinical benefits should be supported by clinical data, there are indirect clinical benefits that can be demonstrated by other evidence, and that could include things like preclinical or bench test data, real-world data such as registries or information derived from different databases or records, as well as data from another IVD that's used with your subject IVD, which does have direct clinical data. So, what are your intended clinical benefits? And just as importantly, what are the reported risks, which will also be included in your risk management files? This helps to illustrate that there are multiple moving pieces and different reports, and that's going to become clear fairly early on in your writing process. So first step is to take a look at your IFU and other feeder documents to help you determine your intended clinical benefits. The need to define what that is a new requirement for IVDR. So where do you start? That's where it's going to be incredibly important to have a well-crafted SOA, because the systematic literature review is going to be a very good place to collect some of that preliminary information. The SOA search often includes mesh terminology that will allow you to capture all of these relevant clinical practice guidelines. And ad hoc searches may still be necessary to support this, but ultimately you want to ensure that you've identified meaningful performance objectives. They must be related to the IVD, and they must be measurable and clearly defined within the guidelines and the recommendations. So you may have to get a little bit creative, and you may have to pull from a lot of these articles that you've been reading as part of your review in preparation for writing this SOA section. This is going to help you, and it may be sort of an iterative process as you're trying to establish what are the most commonly reported, as well as the most relevant and measurable performance objectives that I should be selecting from. So really, until we start to receive feedback from notified bodies and we get a more complete picture of the expectations for each of these different reports, we're going to rely on our extensive experience writing CERs for our medical devices. The Performance Evaluation Report, or the PER, will verify safety and performance of the IVD. And that will include a detailed assessment of the clinical benefits of the IVD when used as intended. To accomplish this goal, you must define state of the art and explain how this specific IVD technology compares to similar technologies within the diagnostic landscape. That's the purpose of the SVR, which we're talking about today. But together, the SVR and the CPR will seek to define and justify the safety and performance measures by which you will judge your IVD against similar competitor devices. The competitor data from the SVR is therefore a critical input for many of these other reports, including your post-market performance and surveillance plans, because it helps to justify the choice of the safety and performance objectives. But it also helps you establish what is the acceptable safety and performance range for particular measures. One aspect of the SOA section that comes as a surprise to many of our clients is that the data presented here is not data on your IVD. The SVR is establishing the competitive landscape. So you will be presenting a thorough assessment of the performance of other IVDs, not your own. But hopefully this schematic will help you see the complementary nature of each of these different reports. And it will also illustrate the need to carefully choose these safety and performance objectives very early in the IVDR process. These objectives will be discussed and justified at multiple stages along the development of the PER. So if you make changes to these objectives late in the process, it can have a ripple effect, and it will inevitably lead to rewrites or edits or delays. Organization is key to keeping your PER preparations on track, and the SVR is a solid roadmap that will help you identify gaps in your objectives before they can carry through to other aspects of the project. As you develop the SVR template, you should be thinking about not only what types of data you're collecting, but how will you connect that information from the SOA background section to support and justify the choice of objectives and data. while the SOA and the competitive searches are separate and they each have their distinct purposes. They must be complementary in order to support your choice of performance objectives. You need to identify data that validates the overall scientific validity of your IVD technology. And it should also support your assertion that the subject IVD is capable of providing an accurate, state-of-the-art clinical diagnosis based on your analysis of the competitive landscape. So I like to equate the SOA section as sort of a funnel. You're going to start very broad and you're going to discuss the clinical medical condition for each relevant condition for which your IVD is designed. This will include things like the epidemiology, the clinical course, and any description of the medical condition. But that will be a very high-level discussion. Now you're going to step into discussing alternatives, and these can range from very conservative pharmacology, any type of conservative approach or diagnostic, but it can also then lead into some of these more invasive, more precise, specific diagnostics. So this is where having a thorough understanding of the guidelines and the standards will definitely help you. But once you get into this next level where you are identifying the position of your IVD within the entire landscape, that's where you want to include specifics on the performance and safety endpoints. That will be where you'll actually extract the data and develop it into a readable, synthesized format. So as you're developing the SVR template, think about how you're going to connect the information from each of these different sections in order to justify the choice of competitors and performance objectives. These are two different searches and they have very different purposes, but they must be complementary. And that's going to help you establish the scientific validity by providing the evidence that you need to establish that this type of IVD provides an accurate, state-of-the-art clinical diagnosis. The competitor systematic literature review will use very carefully crafted search terms and filters because the articles you're retrieving in this search must be carefully and methodologically soundly screened. You have to be able to define very clear inclusion and exclusion criteria, and then document the articles that you've included or excluded during this entire process. You must show that they were assessed and appraised based on the same set of objective, unbiased standards. This screening and appraisal process must be reproducible and should be justified within the SVR to establish that you've presented a robust and an unbiased approach to identify articles with relevant performance data. We've covered systematic literature review quite a bit in some previous webinars. So again, I encourage you to take a listen to some of those other recordings. But systematic literature review itself has long been the gold standard for evidence-based research. We use it to develop these clinical practice guidelines or recommendations, and also to describe and support academic-based research initiatives. So at its core, the purpose of a systematic literature review is to ensure that you have identified relevant scientific data through an unbiased, methodologically sound process. So this is not an optional exercise. Systematic literature review must be included as a part of your SVR and your SOA section, but it can be an extremely daunting process. So the way we begin is with the help of a qualified reference librarian, we design a search using proper mesh terminology and a robust search platform that allows us to search across multiple databases simultaneously. That strategy is critical for that next stage, which is screening, because each article that we've identified in our search must be screened for relevance based on the inclusion and exclusion criteria that we've selected. So a poorly designed search could easily result in thousands of articles being retrieved. And that means that you could spend a significant amount of your time unnecessarily screening all of those articles, and that will result in a lot of off-target hits. Now, the next step after that is the data extraction, and that's the stage in the process in which you've identified articles with specific data that's relevant to your pre-established performance criteria. In many cases for IVDs, the performance criteria will include things such as sensitivity, specificity, false positives or false negatives, or a limit of detection. So you want to be able to present this data that's been extracted, but you also need to document and present the process by which you arrived at this data set. Okay, and that brings us to the final step, which is the data presentation. So how do you plan to extract, assemble, present, and ultimately synthesize the data that you've collected to arrive at your goal, which is acceptability criteria for your subject IVD? And this is where I'd like to present an example just with something that we're all probably familiar with, a typical dessert menu. So suppose your IVD is ice cream. Your dessert alternatives that you would discuss in part of the SOA background section could include things like cake or pie, but those are just that, they're alternatives. If I order ice cream and you order cake, could I compare our satisfaction ratings? We could, but that would be a very high level comparison because cake and ice cream are not direct comparators. So here in this final section of the data extraction as part of your competitive landscape, you want to present the data for your direct competitive IVDs. So yes, the SOA will describe these other alternatives, and it may present situations in which cake is preferable to ice cream. But if you're establishing the competitive landscape for ice cream, you wouldn't want to compare yourself to cake. The standards are completely separate, and the range of acceptability is also totally different. As an ice cream company reporting on the length of time to melt, would you want to compare melting time of chocolate versus vanilla or brand A versus brand B? Or would you want to compare the melting time of chocolate ice cream to cake? Clearly, cake is not a direct competitor with regards to that particular performance criteria, and your time to melt would not appear to reflect a state-of-the-art technology if you compared yourself with cake. So if you don't select the appropriate competitors, the acceptance measures that you report may not accurately reflect the performance. As you can see here in the table, there are several different brands or several different competitors that you've identified. You will include, often in a tabular format, information about the different performance objectives. You're going to collect the data and then calculate an acceptability range based on the data that's been reported in the articles that you retrieved as part of your competitor systematic literature review. That will be the range by which you will then judge or compare your IVD as part of the PER process. So this data extraction and this generation of this acceptability range relies heavily on a well-crafted systematic literature review for your competitor devices. Okay, so as we're concluding here, we hope that you have a better understanding of, first off, how to assess your SVR project for any unanticipated roadblocks or delays, in the context of now knowing how important and how relevant that SOA will be to each of these different steps along the PER process. We also hope you have a better understanding of why systematic literature review is such a critical tool in this entire writing process. We hope you have a better sense of what information to include within the SVR template, and also where you can actually identify some of these relevant performance objectives. We hope you have an idea of the types of data that should and can be included, how to extract that data, and we will talk more, I'm sure, in the question and answer session about the appraisal process for that competitor landscape. How do you actually assess what can be considered to be a high level of evidence that can be included within the competitive landscape? And then finally, we hope you have a better understanding and an appreciation for how indecision or incomplete supporting documents can negatively impact the SOA, and that will ultimately affect your quality and your timelines.
With that, we'll go ahead and get to our first question.
So the first question is, can or should elements related to the technology, such as the technology used, be taken into account in the scientific validity? And the short answer is absolutely. If you are comparing your test, let's say it's something simple like a pregnancy test, there are multiple ways of diagnosing pregnancy, but you will want to explain the technology or the platform by which your particular testing modality works. You would want to make sure that you are explaining how the technology works and why that can be considered to be a scientifically valid approach to making that particular diagnosis.
All right, great, thank you. Let's go ahead and get to our next question. What is the criteria for grading or rating literature articles?
Okay, so we got a lot of questions beforehand as people were registering regarding grading or criteria or appraisals. There are some really helpful references that I would like to refer folks to. So I'm actually going to put that in the chat so that you have the names of these. The International Medical Device Regulators Forum, or IMDRF, has put out a guideline that uses specific appraisal criteria. And that's one that we've used quite a bit. It will give you a sense of the suitability and as well as the data contribution. So this will look into things like, was the IVD like actually used, which IVD was used? Was it a comparator versus your actual subject device? Was it were they all used with the same use, or were there any deviations in the protocol if they used a sample that was refrigerated and yours is only approved for fresh sample collection? Those would be things that you could account for within the appraisal criteria. It'll also discuss things like the patient group or any particular deficiencies that may have existed as far as the reporting of the data. And then we'll look at some of the different outcome measures or the follow-up period. So together, as part of that grading or that rating, the ratings that we're collecting, we're trying to establish that we have, first off, appraised each of the articles that we've included as part of our competitor landscape, but that we're also taking into account the level of evidence that they are providing. So if it's a level 1, meaning it's a very high level, it means that you have identified data on your subject IVD. It was used as intended in the proper patient group. It was a high quality article with very high quality data. Level 2 may include information regarding off-label usage or patient groups that are very specific or precise, but they still may be included in some instances and still relevant. Whereas as you get further down into the actual, the weighting of these different data sets, you may have information from similar competitor devices that were used on or off label that had deficiencies in the study design or the outcome measures. And so by providing not only the appraisal, but then also the weighting of each of these different articles that were included, that gives you a very good reliability that the data that you are using as part to establish your acceptance criteria are high level and that they're very high quality data sets.
Great, thank you, Sarah. Let's go ahead and get to our next question. Here it is. Can you speak a little bit about the difference between the state of the art in technology versus medicine?
Okay, so I think this one is kind of getting at maybe the use of some of the different software or different platforms compared to an actual, like a clinical test, like a diagnostic tool, like an antibody. So the software to detect an antibody versus an antibody. In both cases, that first beginning section of the SOA is going to be pretty similar. You're still going to need to present on the who and where and why that particular technology would be used in a clinical setting and what its role is in making that clinical diagnosis. Your main place where it might diverge then is in terms of the clinical recommendations. You may have to go into a little bit more detail about the type of technology that you're using and how it compares as far as the accuracy to other technologies that could also be used to make the diagnosis. So let's say, for instance, you're comparing COVID tests, and you've got some COVID tests are doing ELISA based, and some are RT-PCR based. You would, if you are doing an SVR for an ELISA, some ELISA reagent, you would have to describe that technology and then compare yourself to other ELISA-based testing modalities. You wouldn't want to compare yourself directly to a different technology. So it's all going to come down to sort of your specific situation and properly identifying that difference between those alternatives and those comparators or competitors. Those are going to be, that's going to be the main point that you will want to make sure that you're paying attention to. But in large part, the beginning section of the SOA will be relatively similar.
Great, thanks for that clarification. Let's get to our next question. Here it is. Can you expand more on how to select the safety and performance objectives for the SVR?
Yes, so in the case of the SVR, we kind of focus in on many of these performance objectives. You want to have a way to directly compare your IVD with a similar IVD or a comparator. So as you're trying to identify those objectives, the best place really to start is with those clinical guidelines or recommendations. As you're looking at how a particular diagnosis is made, you're going to identify what is the range of acceptable for different classes or different groups of diagnoses or diagnostic tools. So I'm trying to think of a good example, but I guess going to sort of like antibodies. Let's say that you are you have an antibody for a particular cancer biomarker. If you were to identify performance objectives for that biomarker, you would want to identify how similar antibodies that are sold by other companies that are targeted towards the same particular biomarker, how do they judge themselves within the literature? So are they looking at the dilution range? Are they looking at the sensitivity or the specificity? Do they have a particular level of detection? A lot of those things are going to be included within those clinical guidelines and recommendations in terms of this particular type of diagnostic tool provides this rate of false positives, whereas a different tool may provide a higher level of false positives. So you're going to take into account sort of the pros and cons of the different tools as you're reading the clinical recommendations and the guidelines. But then in terms of actually selecting what are these performance objectives, you want to think about which of these objectives have a direct impact on these, the accuracy and the overall scientific validity of your particular IVD.
Great, thank you. And I want to remind everyone to keep sending in your questions. We have about 15 or 20 more minutes to get through them. So if you haven't already typed your question into the Q&A, you should do that soon. Our next question is an interesting one. If our NGS assay reports on greater than 300 variants, does this mean we need to provide literature searches for all variants we provide info for in our report?
I think in this instance, it would really depend on sort of the overall the scope and the scientific validity. So let's say your assay is reporting on that, again, using the tumor biomarker, if you're looking at particular variants that are sort of commonly observed within a particular gene. So I don't know if this is looking at the variants within a single gene, like if you're looking at over 300 BRCA variants in this particular sequencing assay, or if it's scanning across multiple genes. I think in this instance, you would want to include the terms for the different variants as part of your literature search, but know going into it that as you are identifying your comparators, Some comparators may not overlap entirely with every single variant. So you will want to be very careful in sort of documenting that this particular competitive assay only covers 200 of the 300, or sort of what the degree of overlap is, because there may be some variants that are more difficult to detect, or more rare variants. But in general, the technology, the approach is going to be very similar to as if you just were detecting a single variant in your particular assay. You're still going to want to explain sort of what is the overall purpose, when and where and why would your particular assay be used on a clinical sample, what types of clinical samples it would be used with, and the types of information. What is the benefit as far as the information that you would garner from this particular assay? So I guess sort of it's kind of a long answer, but it really will depend on kind of how the assay itself is designed. And that kind of gets back to that, the IFU and sort of how you are defining the purpose and the clinical benefits associated with your particular assay.
Great, thank you for that. Here's our next question. Is the state-of-the-art search used to cover all three parts of the report, clinical, analytical, and validity, or are these separate searches?
So if you think of the SOA as kind of this, it's the link or the glue that's going to tie each of these different reports together. The state-of-the-art is there to serve a very particular purpose. That part of that search is going to give you the background that's necessary to explain the rationale for each of these individual reports. So in each case, if I was describing just the analytical sensitivity or just the clinical setting or the scientific validity, they all have different goals ultimately. The scientific validity is trying to establish that association with the analyte and the disease. The clinical or the CPR is going to describe sort of how that particular diagnostic test performs in a clinical setting, whereas the analytical is going to describe the specifics of your particular assay, the data that you've collected. So it will be one search. You will just have one SOA search, but it should include properly crafted search terminology that will help you then explain the background information that's necessary to link all of these different reports together.
Great, thanks, Sarah. Here's our next question. If the SVR is written early in development, but development is delayed, is an update expected prior to submission?
I think this is definitely something that you would want to discuss with your notified body. But in general, if you have performed the SOA search and you've developed a lot of that medical background, in theory, a lot of that has not changed in recent months. But the competitive landscape may have. So in some cases, we have performed what we refer to as a gap search that can allow you to use the same terminology and the same systematic literature review approach that you conducted when you first wrote it and provide that update so that as you are preparing to submit, the SVR is not substantially behind where the rest of the documents are. You want to make sure that they're representing a similar time frame. So it really will depend on sort of what you mean by delay and sort of how long that particular delay is. But it is quite common to do these gap searches in the event that there is a fairly substantial delay, like on the level of several months, then we do those quite a bit.
All right, thank you. Here's our next question. Do you have any recommendations for literature appraisal system or criteria?
Okay, I think that kind of gets back to the one that I posted in the chat box, that IMDRF. That's the one that I have the most experience with using, and I find that it's very intuitive. You can use a chart or a table to help you explain it, and it's very widely accepted as a versatile appraisal system. And it also does, by having just sort of a 3-level grading system for the level of evidence, it's not overly complicated, and it can generally apply to the vast majority of devices pretty readily.
All right, thank you. Here's our next question. Does literature searches for SVR cover both analytical and clinical performance information of the comparator IVD?
That will really depend on how you frame your safety and performance data. You would not really, within the SVR, your focus is very specific. You are trying to identify the performance and compare the performance of different comparators that utilize a similar approach to your IVD. So you're trying to say, here's the range, here's the acceptability criteria as far as the performance of this type of IVD or this group of IVDs. Once you get into sort of the clinical performance and the safety data, that's where the CPR will really come in. The CPR is where you're trying to identify information relevant to the actual performance of this device or this particular IVD from the patient's perspective. So that's kind of how I tend to think about the data bucket, so to speak, is the SVR is covering the performance of the test itself. How well does this particular type of test perform? When you're looking at the CPR, that gets into more from the patient's perspective, not the perspective of the test itself. So what are the overall risks and benefits to the patient based on the performance of this IVD? And then the analytical performance is going to be, the APR is going to be where you're really going to provide that detail about the overall performance of your particular IVD. So it will definitely tie back to the data that you're presenting within the SVR, but you should definitely expect to perform additional systematic literature reviews to help you support the APR or the CPR to be able to fully capture all of the data based on the purpose of each individual report.
All right, thank you. We have about 10 minutes left in our presentation today, so if you haven't already asked your question, go ahead and send that into the Q&A as soon as you can. Here's our next question, Sarah. How do you deal with specific performance data of competitors not being available during your search?
This can definitely be a challenge. It really will come down to expanding the source of the data, or what are the particular performance objectives that you're collecting on. And this is assuming that you have direct competitors, that you've properly identified that there are competitors available. So it might just be that your particular competitor or your class of IVDs is not well reported within the scientific literature. So you could, in part, identify additional performance objectives by sort of scanning through the literature that is published on your competitors and identify how do they define the performance, how are they grading themselves, what types of data are classically being presented. It may not align with some of the clinical guidelines or recommendations, but it may give you some insights as to sort of the limitations of the data that's available to you. You will also want to try to make sure that you revisit who you define as your competitors. In some cases, there just might not be enough data out there. So you could also go back later and expand your search time frame to include more historic data from when that particular analyte was first identified and sort of how other groups provided data relevant to that particular analyte early in its development. So yeah, it will definitely depend on your particular IVD, but this is not an uncommon problem. It really does help to have that relationship with a resource librarian who can help you really craft the search strings so that you can really truly capture any data that is out there.
Yes, definitely. And if you need help with that, we can definitely schedule an appointment with you. You can reach out to us at consult@criterionedge.com, and we'd be more than happy to discuss that with you.
Yeah, the librarians are your best friend in these cases.
Here's our next question. It's kind of a long one, but I think a good one. We have higher risk tests and devices and need to update our PERs and CERs annually. Do we need to provide a Prisma report of only the new literature that's been published and appraised by us throughout the year in between updates, or do we just keep adding on to our original Prisma report?
This will depend on what your notified body sort of expects. In our experience, the way that we typically handle this for a CER would be to provide a Prisma report for the data that's currently encompassed in this particular update. So whatever is, if it's being carried over from a previous CER, you can include the Prisma from that prior CER as part of an appendix, or you can refer to it. But generally speaking, I wouldn't just continue to tack on year after year, because eventually these Prisma reports are going to get pretty unwieldy. You're going to want to make sure that this is the documentation of how the search was conducted for this particular draft or this particular version of the report.
All right, thank you. We have time for just a few more questions, so if you haven't already, keep sending them in. Here's our next question. How do you approach the state of the art for general use IVDs that are not specific to a particular analyte?
Yes, this is one of those situations where I think it is going to really depend on the particular IVD or the particular I'm trying to think of another good example for this one. Let's say it's more of sort of a general, if it is a general use, if it's something that's used in multiple different types of tests, I think in that instance, you would still want to take a similar approach. Why and where is your IVD used? Is it used in a particular type of sample? Is it used for a particular type of test, or is it specific to a certain class of tests? Are there any unique situations in which it's better or worse than alternatives? So I think you can still take the same general approach. The establishing the scientific validity of the association of the analyte to the medical condition is going to be where you would have to really sort of think outside the box. You're going to have to make sure that you're clear in how this particular IVD supports the scientific validity of the test or whatever that test is hoping to accomplish in terms of the diagnostic capabilities. So yeah, without knowing any specifics, that would be my approach. I would try to take the same general approach as I would regardless of what the device is, having that sort of very systematic, that organized template that you can then adapt to specific unique situations. The template is going to be your roadmap. So having a template that's been tried and trusted and used before for other devices or other IVDs will be infinitely helpful rather than trying to sort of craft this whole story from scratch.
Yeah, thank you for that. Here's our next question. Where do you best look for performance information of competitors?
So in many instances, you can find this by doing, if you craft your systematic literature review and your search terminology to capture the names of the competitors, you will hopefully identify articles that contain extractable data. So they may say that in this particular test, and often it's not the competitor, the test itself, that's the focus of the paper or the focus of the article. It's sort of used incidentally as part of this overall article. So it's not necessarily going to be like, here's a test on this particular, like, here's us trying to validate this particular type of COVID-19 test. It's going to say, no, we were testing for COVID-19. We happened to use this test. Here was our accuracy. Here were our sensitivity and specificity. So again, it goes back to that search terminology and the search strings. if you can use a platform that supports advanced mesh terminology, something like a ProQuest dialog, rather than just doing a quick PubMed search that uses and/or and leaves it at that, it's going to help you identify articles that specifically call out and mention your competitors without those competitors being the focus of the entire article. So you can get there. It may require some screening. The screening may be a little bit more time intensive if it's a particular type of test that there just is not a whole lot of specific data regarding.
Okay, thank you. Here's our next question, Sarah. For a microbiology product only used for presumptive identification, how would the analyte be described and how would the SVR be demonstrated?
So I'm assuming this is, the way I'm sort of reading it is, let's say this is it's presumed because it's excluding the other obvious sources. So in that case, you don't necessarily have a particular analyte that you're looking for. You're sort of excluding other analytes. That's kind of the way that I'm reading it. So in this case, if your product is designed to state, like, let's say it's trying to detect gram positive versus gram negative, if it's sort of excluding that it's not gram positive, you may want to still assess which analytes it's excluding and sort of how it's going about making that identification. So you'll want to get into some information specifically about how that test is conducted, what's the sort of mechanism behind that test, and then show that this test is sort of how in your competitive landscape, that's where you will show the overall accuracy of this particular test. You're not going to compare yourself to an antibody that's specific for one particular microbe. But if it's a presumptive identification, you can state how well it performs in terms of how many cases does it miss, or how many cases is it successful at diagnosing, and sort of how its exclusion, how is it able to exclude any other particular pathogens.
All right, thank you. We have time for one last question. So here it is. How do you adjust your search terms to make sure the right amount of literature is retrieved? And how would you know if you should keep narrowing down?
I think a lot of this will come back to experience. I know in our instance, we will go back and forth quite a bit with our librarian to really hone in on what's a reasonable amount of literature that you want to screen. If you're doing, depending on your device, if you're doing something like COVID testing and you did just very simple search terms, you're going to pull down thousands of articles in just the past 2 years. And if that's your competitor search strategy, you are then expected to show that you have screened all 1,000+ articles, and that can get very time consuming. So we will sort of craft the terminology and the search strings to include things like the specific device names that we're looking for. You can adjust sort of how close one word is to another word. So rather than looking for COVID and test appearing anywhere, it will look for COVID test together. Or maybe you want to do COVID ELISA-based tests and sort of limit it like that. So you can look for COVID being near or next to ELISA. So there are some tricks, and again, your librarians are your best friends. So there's not like sort of a set number that you're aiming for to get it narrowed down to, but just know that whatever your final number you arrive at is, you're expected to have screened all of those articles and to then present a detailed screening and appraisal and extraction of those articles. So I would just suggest trying, sort of knowing how, being aware that that can be a particularly time-consuming step.
We are out of time. Thank you for joining us today. We have a few more webinars coming up, a part of our Ask the Expert series. Our next one is on September 22nd on the safety and performance measures and acceptance criteria. These are short 45-minute live Q&A sessions where you can ask your questions live like we just did and get some answers from our president, Lori Mitchell. And our next Ask the Expert would be October 6th, and that is on the performance evaluation report. Again, if we didn't get to your question today, or if you have more questions on the content we discussed, you can schedule your free appointment at consult@criterionedge.com. Thank you everyone again for joining us and for your wonderful questions. Take care and have a nice day.